afternoon, everyone. Welcome to this overview a bit about metabolic MR imaging. I will talk a bit what MR can do, realizing well that metabolic MR is not really well suited for metabolic imaging, to be honest, but we will go through that. I will also talk a bit about standard clinical contrast agents, because they're quite linked how the research area of metabolic MR imaging and the development of the contrast agents are developed historically. So my name is Rene. I work at the Lund BioImaging Center. It's an infrastructure for biomedical imaging at the medical faculty at Lund University. We work what we call platforms. So we have a microscopy platform, which is growing a lot. There's a lot of development in the light world, to be honest. Then we have our nuclear platform, which is PET and SPECT CT for rodents, and a dedicated high resolution micro CT for rodents. Next door in the building, we have our rodent MRI 9.4 Tesla Bruker system. And then we go over to the clinic, where we have access to the standard clinical machines at 1.5 and 3 Tesla. Then we have our national 70 Tesla MRI facility, where I'm currently working at, and especially microscopy and micro CT generate incredible large data sets, as most of you probably are aware of. And to handle these and how to present the data, we have a special people that's working on analysis and visualization of these large data sets. LBIC of the Imaging Center was founded in 2008. We have now about 17 employees, and we are tightly connected also to the MAX4 synchrotron facility and the ESS neutron beamline facility that's almost up and running, give it a few years. So what I will do today, I will talk about MRI. But if you talk about MRI, we can do a lot of applications. This slide gives an overview of. It's a lot of applications that we've been running through the years. Cardiac imaging on the mouse. You can look at a human menisc ex vivo and look at composition. You can look at ex vivo rabbit brain, at how the fiber tracking is, developing brain. You can look in vivo on mouse tumor perfusion or stroke models in the rat. And if you go to the right of this side, you see just anatomical imaging of the mouse in vivo or ex vivo rat brain, looking at angiography and, for example, here the use of contrast agents to look at the uptake of contrast agents in the inner ear of the mouse. So there are a lot of diversity, but none of these are basically done with any metabolism. So to be fair, metabolism is a relative small area of the total MR field. So if you just name them as they are, there are different applications. As I said, we can look at anatomical changes, bleeding, inflammation. We can look at flow and diffusion, looking at susceptibility, basically iron contact-driven, brain activity, what's called functional imaging, and at the end we have our metabolism. MR spectroscopy is, as it basically is what it is in the basis, is 50 years old, so there's not a lot new there, but there's some applications that have really more developed the last years. I will shortly touch on deuterium MRI, not a lot, because the time doesn't really allow that today, but I will talk about CEST imaging and show a bit about hyperpolarization MRI and how it can be used. The question that will arise is then, why do we do metabolism? And this is a slide I got from Dennis Klomp from Utrecht in the Netherlands. I think it's a very nice image where you can see CT images before chemotherapy and 1, 2, and 3 months during the treatment. And if you look at the imaging, that you see the tumor is growing, and even after 2 months, it's probably still the same size as before the treatment started. And at 3 months, it's suddenly kind of gone in these images. But we're pretty sure that during these first 2 months, things happen in the tumor. And that's why Dennis wrote here, morphology is too late. And I would just say, well, maybe with metabolic information, we could have seen in an earlier stage if the treatment was successful or not. And I think that's the main key of metabolic MR these days, that we want to identify these patients who are not responding to therapy. So many of the examples I give today involve contrast agents, which is normal for PET and SPECT, because you need your agent for PET and SPECT to work. And if you look at MR, there are clinical MR contrast agents as well to get some extra contrast in disease. But there was a problem around gadolinium-enhanced MRI, and I think it's of relevance to take that into account because it changed the field of research, it changed the field of metabolic imaging. And still today, I think many of these techniques that are used for metabolic MR imaging are kind of an alternative to gadolinium-enhanced MRI. So for those who are not really familiar with that, this is how these clinical agents work. You have a control image. You see on the left, already see here, there's some abnormal tissue. You inject the contrast agent, and where the contrast agent is located, you get an enhancement of the signal. And in this case, it's due to a disruptive blood-brain barrier, and you get some delay washout, and there's a window where there's a clear contrast of the disease. And if you look then what's available clinically, well, this is the whole list. Although they have all different names, they're all based on the very same thing. There's basically only one type of contrast agent clinically approved, and they all, although the formulation might be different, they all do the same thing. But then something happened about 15, 20 years ago, that there were some patients identified with the reduced kidney function, that they got kidney failure. And then it started a complete investigation about these contrast agents. So the result was that 3 of them, which shared a common kind of formulation, are withdrawn from the market. And what happened that, for example, Omniscan, owned by GE, made Clariscan, which was a genetic variant of Dotaram, which was off patent. So the field changed a bit. And if you look today, this is what's available, clinically approved. And as I said, they all are basically the same. Dotaram, Clariscan, Prohance, and Gedovist. And there's 2 new agents approved 2 years ago, which is pretty unique. The first ones in 20 years. They're more effective, and therefore can be used in a lower dose. And the idea is as well as the lower dose, the better in a way, like with any contrast agent. And then we have the liver-specific ones. The reason I show this with these gadolinium agents is that it changed the field, because after this was discovered or realized that there were problems with some of these agents, as that directly the day it became clear, and I was working for GE at that time, is that all contrast agent research for MR was cancelled or on hold, just vanished from the radar. And it also means for the authorities to approve a new agent is getting more difficult, because knowing the history of gadolinium, they are not very eager to approve something new. And that also means it's very, very expensive, and it is just difficult to prove you have the right safety profile for new contrast agents. So there's a line that says, well, we try to avoid them and develop other techniques. One example is APT-cyst, for example. And if you still have to inject something, well, let's use something that's already proven to be tolerated or approved for the human body. Manganese is known to be tolerated well, so it works similar as gadolinium, and there are still companies around, even close to me in Lund, that develop agents based on manganese. Glucose is well tolerated. You take what they use, the glucose tolerance test in the later phase of pregnancy. Take that kind of dose and drink it, and you can do MR with that as well. So people try to use glucose as an alternative for gadolinium. You can use, and then we get more into the metabolic imaging, use carbon-13 labeled compounds. Again, glucose comes back, but even pyruvate and fumarate, who are documented to be relatively safe. Deuterium labeled compounds, and again, same compound, glucose, just in a different way. And the latest thing I think this says is that they have seen that these X-ray iodinated agents that are administered using for CT at very high amounts are also can be used in MR. So again, you use something that's approved in order to be able to get approval also for MR agents. But what about the sensitivity then of MR and PET? And I think it's very important to put these two in perspective. MR is not a very sensitive imaging technology. The images are really cool, but MR in itself is not sensitive. And the difference with PET is pretty drastic. It's about 100,000 to 1 million times more sensitive than MRI. And then if you look at contrast agents like PET FDG, it's about, if you do the math, it's about 1 nanomolar in solution. It's very low amount. It also means that safety profiles are way more easy to deal with than with MR. Gadolinium-based agents have about 0.5 molar concentration, so it's a huge difference. And if you look at the signal we normally get in normal imaging in MR, and you're looking at the protons from water and fat, water is about 80 molar in concentration. So it's a completely different ball game that MR is using, but the combination of high concentrations of water or higher quantities of water still saves the technology in a way. If you want to do metabolic imaging with MR, the largest one in the brain are 1 to 10 millimolar, talking about N-acetyl aspartate, creatine, glutamate, glutamine, taurine. None of them are proven to be a real good biomarker, but these are the higher ones. And the real interesting ones in the micromolar range or even lower are out of scope for MR. So the question is, can we do something about this and increase the sensitivity of MR just to make a next step in detection of metabolites? Well, you can buy stronger magnets. That's what people do all the time, and it benefits the metabolic imaging a lot. So what you do, basically increase the polarization, and what it means I will come back to. And I will explain what then hyperpolarization means, which is a situation created in another machine. But the term polarization I'll come back to. What we also can do, we can use the strong water signal to detect metabolites that are in exchange with water. So we can, if you have a component that's just communicating with water, and even though we don't see the component, we still can look at its effects in the strong water signal. That's what CEST is about, and I'll come back to it in a few minutes. It's called saturation transfer. It's a technique that's also 50 years old. It's used a lot in spectroscopy. It has been rebranded and reused and given a new acronym. But in the basis, it's a very old technique. Even MR spectroscopy got revised. We call it now a metabolic MRI. It's the same thing that people are doing for 50 years. It's just that now we have higher fields. It's clinical use for 7 Tesla. That suddenly opens up for new possibilities and the technology also has been approved a lot. So we're just moving the borders all the time. As I said, we'll talk a bit about Karma 13 with hyperpolarization, which boosts the signal dramatically, and a bit about the deuterium imaging that will help us. Before I can go in that, I'll take a very short crash course in MR spectroscopy. So if we look at the signal we get in MR, we get the signal from protons, the protons from water and lipids. So these blue dots over here. And that's the signal generating compound, you can say. So if you look at an image, and this is an image of the upper leg, you can recognize hopefully the muscle. In the middle, you see the bone, which is black. We don't see that on MR. I'm seeing the bone marrow bright. But if you look superficial, you have a lot of superficial lipids over here. And if I would pick a pixel in the middle of the muscle, I would I will see a signal for water, and I will get a signal for lipids. And I will move my pixel to the edge where I know there are a lot of lipids. You can see these 2 peaks change. The water is low and the lipids go up. So, and there is a scale below here. And this is what we call an MR spectrum. It's like the normal, if people know familiar with NMR, it's the same thing. You get an MR spectrum, and the frequency a substance is resonating in is also identifying the peak. So we can use it to fingerprint the metabolic composition. So in this case, we see only lipids and water. And that's what you're going to play with. So I'd like to introduce now the concept of CEST imaging. Again, we have an MR spectrum. We also have 2 peaks here, not water and fat, but let's call it water and a little other substance we have, either added or present in the human body. And what we can do MR is that we can use a so-called saturation pulse. It means that you saturate the signal, and it means in this spectrum presentation, the signal will be disappearing basically. But if these signals which are invisible are communicating with the water, it will be transferred to the signal, and my water signal will go down. And if there's no exchange between those two, the water will be unaffected. So in A, we know we have exchange, and in B, there's none. And that's basically what we do in these so-called saturation transfer techniques. So if we have again this example here, the small peak and the water peak, I represent this little peak with 5 dots here, and I have the water with all these green dots over here. We use our radio frequency waves, and we color them in this case orange, and orange means invisible, so they will be gone. I don't see them anymore. But if there's exchange between those two, and let's say 3 of them move to the right, well, as there are no orange on the right side, we know 3 greens come to the left. And we keep on irradiating with these frequency waves continuously. That means we color them again orange, and again dots will move to the right, and very likely greens will come back. And you can do this continuously, and after a while, you manage to label a substantial part of the large water signal with orange dots just based on the exchange. And as a result of this mixture, your water signal decreases. And since this water signal is so high, we can detect very small differences in changes. So a percentage can be accurately detected because the signal is so strong. And for those who are familiar with MR, is that this only works when the exchange is fast compared to the T1 relaxation. And what that means is that if I would stop applying this pulse here, everything will be green again after a certain time. It's like a half-life in nuclear medicine with isotopes. Only the half-life in this case for MR is then a few seconds on the proton side. So but if this is going 1,000 per second, it still will work. So that's what is called then saturation transfer, and how it will work in practice is shown in this figure. The reference to the publication comes into the next slide. But what they have done is that they had just a phantom solution with barbituric acid, where you have the MR spectrum with CH2, the water, and the NH groups over here. So now we do that exercise that I just said. We have a saturation pulse set on frequency, and we start pulsing continuously. Well, over here, there's no exchange, so nothing will happen. We can move our frequency to CH2, and they are not communicating apparently because nothing is happening on the water signal. So no protons of the CH2 are leaving the CH2 group and be bound and exchanged with water. Then we get to the water signal. Well, signal is gone, is not really what we want to see. We continue our frequency, and the water signal will come back. And the interesting part happens when we reach the NH group. So suddenly we see that the signal drops because one of these protons are not bound continuously to the NH group. They also will change to the water and forth and back. And it gets really interesting if you realize that this exchange is pH dependent. So if I would redo this experiment in an imaging kind of way, I can make an image of 4 vials. But if I put that frequency pulse on here, exactly here, and look at the exchange with water, it will affect the water signal. And that's what I see here. I have 4 vials that change the intensity, but that exchange is depending on the pH. So basically what I can do pH imaging in a phantom or pH imaging in vivo. But suddenly we open up a complete different way of doing MR, and you could call it metabolic imaging. We do looking at the physiology. So this is an example, and there are a lot of publications with this CES technique. This is just one, and if you're interested, I would urge you to just look in the net or start with Silvio Aim from Torino. He's done a lot of chemistry and built a lot of molecules that are fascinating in his function. But this is an example where you see that you can look at pH in a tumor model in the animals. The disadvantage of this is that most of these studies that are published use a compound that is manufactured in the lab, and it's tolerable. You can use it in animals, but it will never be improved clinically. So it kind of this field is kind of limited to the animal work, because none of these compounds are clinically approved. So if you go to the human case and you want to do a similar technique, you can either, in this case, look something that's present in the human brain, for example, which is known to have an exchange with water, and in this case it's the amide proton, and it's called amide proton transfer CEST. And if you look within the circle, over here you see there, compared to the circle over here where there are more red dots, is that a grade 2 astrocytoma shows less effect than a glioblastoma grade 4. So they can discriminate tumors based on this kind of exchange with water. And this is an early public work, but today for Philips at 3 Tesla, it's a standard product. So it's harmonized, it's standardized, color tables are the same, and everything is it works really well. Another example is using the glucose. You're doing the same thing. You drink the glucose or inject the glucose and look at how glucose and water are communicating. So this is an example from a colleague of mine, Anina, and shows you that if you have a conventional contrast agent, you get these curves where the contrast agent, enters the region of interest. You see the same experiment done with sugar. What you can see at least is that, for example, this curve, the lowest, is the lowest here, and this orange, dark orange, is also the highest here. So relatively, they are reproducible, but you can also see that the glucose experiment is way more noisy, and it's not up to the level you want to reach with contrast agents. But people try to replace the gadolinium. Also a bit driven, it's more recent, that because the gadolinium is in its molecule is non-toxic, but when it's released from its, just as an atom in itself, is toxic. It leaves the kidneys, so at the end of the day, it ends up in all the oceans of the patients, and they see a lot of animals now that also accumulate gadolinium in nature and shouldn't be there. So there are other forces except for patient toxicology, then it's also just an environmental issue. So to summarize now, a bit about CEST. So the fundamental difference is that if you look at gadolinium on the left side, it's there when you inject it, and the CEST it's only there when you try it in the MR sequence to enable a certain feature. So if you inject gadolinium, you always have to do as the last scan if you want to do other anatomical imaging, because all the images will be affected. This can be done at any time, and if you would inject a cocktail of components, you can play around with this to visualize different physiology. But again, this is limited to animal work. There's nothing done in the human case. So back to the spectroscopy. So far, as I said, the signal is generated by the protons in this water and lipid molecule. But there are different nuclei we can look at, and that's what's called the multinuclei, and this, basically it's in this field where the metabolic imaging is, I would say, getting more interesting. This table is a lot of values, and if you're not familiar with that, I would just ask you only to focus on the frequency for now. So each nucleus that we can measure has a specific frequency it will work on. So at 3 Tesla, the protons of water resonate at 128 megahertz, while at 70 we're at almost 300. And you can see these frequencies change depending on the nucleus we want to look at. And that means that you could draw a large spectrum, which I got from Dennis here, where you could start at water on the left, and while lowering the frequency, you will pass all these nuclei that can be used in what they call metabolic MRI these days. The problem is we cannot use them at all at the same time because we need dedicated hardware. The amplifiers currently these days are broadband, so that's not really an issue. It's that what we call our radio frequency coil, that's basically used to do the scanning. In this case, is for a rodent, and if it would be human, you could think of maybe stick your arm in this, but depending on where you want to measure, you have to something be around, which is called the radio frequency coil. And you can see in this one, it says 2 frequencies it will work on, the proton 400 and fluorine 376. So what's a bit limiting, if you want to change that, you have to have dedicated hardware for RF calls for every nucleus. So I will talk a bit about 3 nuclei, the carbon-13, the deuterium, the 2H, and the phosphorus, 31P, and start with the carbon-13. So I asked you to look at the frequencies, which are lower. And if you now look at this column, then you see its natural abundance. So if you take a lipid with all these carbon atoms in it, only 1% is carbon 13, majority is carbon 12. That means that, yes, we can look at the carbon signal of lipids, which are a high abundance, but it's a challenge to get carbon 13 of glucose, for example. So what you could do is that you take glucose and change all the carbon 12s with carbon 13, and suddenly you increase your SNR of your sensitivity for this carbon 13 with a factor of 100 almost. And suddenly these compounds can act as a tracer molecule, a contrast agent, if you would call it. So carbon 13 studies to visualize metabolism or general carbon, even carbon 14 are being used a lot. You have carbon 11 variant in PET, so it's a very often used molecule to study metabolism. The trick here is that the combination with hyperpolarization. So we use the carbon 13 technology around for decades and add something to amplify the signal dramatically. So for that, I have to introduce to you the concept of polarization. It's basically said, we could say it's just a signal available in the MR machine. So polarization is looking at the difference in the quantum mechanic description, spins up and down, and the difference between these two is how much signal you have available. And if the stronger the field you have, the more you will drive this equilibrium to one side, basically. This equation here is all constants, except for the magnetic field you're working at and the temperature. So the temperature we cannot do a lot of because we're working with living samples, so you cannot cool it down to 0 Kelvin. It's really limited. So in practice, the only thing we have to change this polarization is to buy stronger and stronger magnets. So that's what people are doing for many reasons. But it's also good to realize is that these, even at 9.4 Tesla, this equilibrium change is only 1 ppm. This in practice it means that 1 out of a million is the difference. It's really small. It's not that you're fully charged when you're lying into an MR system. It's a very small difference. Still large enough to be detectable, but it's small. So hyperpolarization then is a process where you drive this to unity. And the equivalence would be a magnet of maybe 1,000, 10,000 Tesla, which doesn't exist. So the technology I will talk about, the hyperpolarization, is an artificial state created outside normal MR scanner. So if you look at the very first prototype here from the group of Malmö, South Sweden, it's an engineering project. Vacuum pumps, you have to get it under low pressure. You're working with liquid helium. You work with microwaves. But at the end, if you get the sample out of this system after a while, it's proven to work because this is a spectrum, almost 3 days of scanning with this signal to noise, and this is a single scan. So it's a dramatic increase of signal to noise. And how it would look in the lab in the early days is that this is a 11.7 magnet, vertical system. This is what we call the polarizer. It's also a magnet. This one's at 5 Tesla. The very first one's at 3.3. But the message is that this is the machine producing the contrast agent. So once you take it out, you can transport it to the magnet, add it to the cells or whatever you want to do, and do your studies. But this process had to go fast because I said a bit also with the CEST imaging with the green dots that, yes, they are orange, but they have a limited lifetime, the T1. They all become green again within a few seconds. And this is exactly the case here because I have this huge signal with 10,000 more signal than normal NMR, But if I do nothing, it will just go back to the thermal equilibrium. And this time here is that what we call the T1 relaxation, as I said before, and for protons it's only a few seconds. So it will not help you a lot. It will work in a lab with a vertical system, but if you want to use this as a contrast agent in human use, well, the time it takes from the injection site to the organ of interest without even considering any metabolism is already way longer than the half-life of this component. So for protons, it won't really work. So that's one reason it came out at carbon-13. If looking at the C1s, they have about a half-life of 40 seconds. And they had an advantage. There's a huge literature around it using the metabolic study. So those two things came together. With the T1 about 40 seconds, you have 3 times this value, about 2 minutes to work. And as long as you start with a very high signal level, that hopefully there's enough left at the end. That's kind of the idea. So it's almost like the equivalent with radioactive oxygen, which also has a direct transfer line from the reactor into the patient because the half-life is so short. If you look at pyruvate, which was the first one to see the light, it has different metabolic fates if you inject it. So it goes to lactate or alanine in the glycolysis. It will enter the citric acid cycle and become acetyl-CoA, and there's a theoretical pathway to oxaloacetate, but I've never seen that showing up. So how does it look like a signal? Again, we have an MR spectrum like I showed before. It's kind of a different scale here. We have a different frequency. But what we look at is this red carbon-13, and it has this unique position over here. There's a different form of hydrate, a different shape of this molecule. It's just there. It's background, let's call it that. We ignore it for this presentation. If this pyruvate is transferred into lactate, you see the carboxyl group is still the same, but due to this change in structure next to the carbon-13, its frequency in the MR shifts a bit. So the lactate has this unique position in the MR spectrum over here. The same thing we can do for alanine, and we'll end up somewhere in the middle. And for this specific label position, we will lose our label in the decarboxylation of pyruvate into the citric acid cycle. It will become CO2, and we'll find it as bicarbonate in the tissue or in the animal. And the bicarbonate has its resonance over here. So this is kind of the game we're playing with. We inject the green contrast agent, and during the time, signals for these other metabolites can or might be formed. So and that's also the difference between PET and carbon-13. Because for PET, we will not be able to discriminate these three. We'll just look at a total signal. MR had the advantage that we can tell us what the total signal is, also which metabolic pathways it has taken. But still, I mean, as you would say, sensitivity difference. So this is the very first example that's been used to show the possibility or the feasibility with pyruvate. We used a very thick slice around the spinal cord of the rat. So this is spinal cord, these are the muscles, this is stomach side. Very thick slice. So it's a projection over this volume, but we can get away with that because the muscle is pretty uniform in thickness over this area. And if you do this experiment, you can see the pyruvate coming into the large vessel in the middle of the animal. And at the same time, you see that the lactate, but of all, the alanine is formed, being specific for the skeletal muscle around the spinal cord. So pyruvate injected, and these are the products imaged out of that. This will not really take off. So if you have to go to the disease model, and this is for cancer, where we have an animal model, where a rat model, there's a tumor over here. And again, the same thing as we had for the muscle with the lipids. If you would take a pixel somewhere in the middle, you would hope that we only see pyruvate. And if you would take a pixel in the tumor, that we see the lactate. And in that sense, you would have an imaging biomarker for cancer. And that worked really well. These are the images of these publications where you can see that pyruvate has several hotspots in the animal. You see again alanine being rather specific for the muscle, and you see the high intensity in the tumor in this example. And this was proof enough at this time, because this was part from General Electric, that to say, well, this is something we believe in, this might work, let's go into development. And that this decision is taken in 2004 or 5, so it's quite a while ago already. And people are taking off, and it's been a long journey to go to clinical research. I showed you the very first example of that polarizer in the lab with all the cabling. You cannot have that in the clinic. So these days it looks like a fridge, and there are other companies now building even smaller machines, faster machines. But this is how a clinical approved one looks like. Since we have to produce the contrast agent next to the MR or close proximity because of this half-life of these agents, they also mean you're producing a medicine on site. So everything has to be GMP. You have to have a pharmacist that releases it to be safe. Then you have to run to the MR and do the scanning. At that time, the technology became clear. There were no coils that were used for carbon-13 because no one used it in a clinical setting. It requires a different set of sequences that can handle hyperpolarized signal, but also image different metabolites at the same time. Old school spectroscopy is very inefficient and way too slow. So a new library of sequences have been developed specific for this application. And you have not to forget also the whole data analysis of all these data sets. So this has taken 10, 15 years, and this as well. So I don't say we are already regulatory approval, but there are clinical trials ongoing. So I have 2 examples on that. This is an example from the group of Cambridge. I'm not right. I'm right. If not, I'm sorry for that. Just 2 genetic backgrounds of breast cancer, where you look at the ratio of lactate over pyruvate after you injected pyruvate itself. And you can see in this figure on the right is that there's a huge difference in response, at least in this ratio, between a responder and a non-responder. And again, it goes back also to the earlier thing I said. It's very crucial to be able to identify a patient under treatment that's not responding to therapy. So this might be a way to look into that. And there are a lot of techniques currently to do that in research. Another example is in the front page of Radiology 2 years ago. This is definitely from the Cambridge group. Human brain for glioblastoma works really well, injecting pyruvate and look at the lactate signals. So it's been a 20 years journey there, but at the end we'll get to the human application. So switch gears to deuterium, the 2H. You will see 2H, and even for me, 2H as a spelling, or deuterium as the D, heavy water, deuterium metabolic imaging, DMI. It's a bit often used. It has a very low natural abundance. So most of the water is just 1H, and the remaining of this not being 100% is the deuterium side. Again, that also means that it can be used as a contrast agent if you would, for example, in glucose, switch all the protons for a 2H group. Suddenly you have a very strong signal with no background signal present. So how is it being used? It's just an example. And again, on cancer, again, focusing on lactate. You see on the left here, these spectra, you see a bit of water signal because, as I showed, not 100% of the water is 1H. There's a natural background of this 0.015. The glucose is on close to that. That's the one that's injected. And you can see that either lactate is produced or glutamate. And the glutamate is the end product in the brain after you gone through the oxidative phosphorylation. So basically oxidative phosphorylation phase. And if this one is hampered or not working optimally, you get lactate production instead. And if you look at the right here, you see these colorful images that, yes, again, large tumor, lactate gives the hotspot, and it corresponds to the area where the oxidative phosphorylation is either shut down or working slowly. The glutamine signal is low. So all the techniques I've shown so far, they focus on the same metabolites in different ways. And at the end of the day, one of them that should, work well and be hopefully clinically useful. So this is deuterium. I'll come back to that in a minute in the context of the European project that Erika mentioned in the beginning, where lactate is one of the biomarkers. I go into the last one, and that's a pretty cool idea, to be honest, done by Dennis Klomp in Utrecht. So what if you take standard 3 Tesla equipment, coils, gradients, amplifiers, and just connect them to a 7T system? Or you say it will not work. No, it will not work for protons, which it has to have 298. But if you go down the list, there's one here, it's almost the same value. So if I would use standard electronics from a 3T clinical system connected to a 7T magnet, I will be able to do phosphorus imaging. So how would that look like in an idea? Very short. Normally you see the hospital, you see the scanners looking like this. But inside this cover, there are a lot of coils. Not really relevant today, but inside this circle, or this one here, there's the gradient coil, which is the one that's causing all the noise, and it's used to position space, coding space inside the magnet. This one. Inside this gradient coil, in all clinical systems, there's an RF body coil, thinner, that's used to sending and sometimes even receiving the signals, just to do creating the images, basically. And until now, all 7T systems on the market lack this coil. So there's some space left. So all these systems, 7T in the world these days, or most of them do not have an RF body coil. That means so you can build your own one. That's what they did. Some picture of the electronics. Again, you see they come from proton 3T to use for phosphorus. Build the coils. You can do it for any manufacturer. And you can do this kind of metabolic imaging at 7T. I'll come back to it very soon because I want to remind you that why do we do this? Because we really think that normal imaging is not sensitive enough. We want to identify these patients not responding to therapy. I think that's where the strength of MR is left, let's say, or PET is maybe not the strongest case. So that's what the Utrecht group is pursuing, doing this with the phosphorus. So since they have this large body coil, seeing the whole human body, they can do spectroscopy over the whole body. You can do a lung cancer. I'll go back here. So I won't explain all the resonances, but what I would just say, the biomarker for cancer is here, this glycerol phosphate, choline and glycerol phosphate, etonolamine. They increase in cancer. So this is a biomarker. So what other ones will go down, but this is the crucial one. And you want to identify that one as a cancer biomarker. I go quickly, and this is a very busy slide, and that's exactly my point. Since we have so much data covering the whole body, and so many spectra with so many peaks that varying, and the handling of that, it's very complicated to analyze this. So that I think currently with all the new algorithms and AI, I think it might possible these days also with the computers that are faster, more powerful, to do this kind of analysis on a large scale. But this is the basis of it. So using phosphorus to detect cancer or to detect maybe a response to therapy. And that's exactly the basis of that European project. This is my final slide. It's called ILLUMINATE. You see the participating partners. Euro-BioImaging is also involved in that, where patients with metastatic prostate cancer, as kind of end-of-the-line treatment, they are at least currently in Germany and the Netherlands, the only 2 countries I'm aware of, are offered lutetium therapy. That means it's produced in a reactor with an antibody specific for the cancer. I think it's the PCA it will attach to, and the lutetium itself with local radiation will destroy the tumor. It's a very expensive therapy, but it's offered to all patients. But they don't know if some people do not respond at all. And that's the field, because the therapy is expensive, where MR can come in. So the idea is that with 7 Tesla metabolic imaging, that we will, using this phosphorus whole scan technique, looking at this marker for membrane building, the glycerol phosphates, and taking deuterium in. So again, looking at the lactate by giving the patients glucose. So you're looking at the lactate on the deuterium side and the membrane building component in the phosphorus side as a biomarker to identify patients that are not responding to this expansor therapy. So it will say if we detect 1 or 2 patients out of 15, so it's a very small number, we save already so much money in this kind of therapy that we can offer all these people a 70 exam. So it's kind of cynical. Thanks to these very expensive therapies, there's suddenly space for MR left for these kind of advanced imaging. With that being said, yeah, thank you for your attention.